Chapter 7, the title is Patterns of Inheritance. So these are the chapter objectives. We're going to learn what is genotype and what is phenotype. And then we're going to learn Gregory Mendel's experiments and some of the laws that he developed from those experiments. Basically laws of segregation and laws of independent assortment. Then we're going to learn what is a Punnett square and how we can use Punnett square to determine or predict the phenotype of offspring. And finally, we're going to learn or we're going to kind of discuss how we can determine or how a person's or individual's phenotype can vary depending on the genetic composition and the influence of the environment. So as usually this chapter also has a background story, and the background story talks about canine phenotype or the appearance of dogs. So here one person is a dog researcher, and then the other person that we discuss in this chapter is a dog breeder. So both of these people, they are dog loving people. So here the main dog or the main furry friend is called Georgie, and he is a Portuguese water dog. So this Georgie, he died from a genetic disease called Addison disease. So this dog belonged to a geneticist, a scientist that worked on genes, and called Gordon Lark. So he was working on a soybean, and once Georgie died due to this genetic disease, what he did was he kind of adopted another same type dog, so from the same Portuguese water dog, and she got from a dog breeder, Karen Miller. So when she provided the dog, she asked Gordon to find out what caused these genetic diseases, or to do some more research on these genetic diseases on furry friends or dogs. So before we go to more details, let's talk about some of the definitions of some terms. So when we talk about genetic traits, so genetic traits are any inherited characteristics of an organism. So like for example, like a skin color, eye color. So those are physical characteristics that we inherit from our parents. So that's what we call a genetic trait. And invariant. So invariant traits are the same in all individuals of the species, and the variable traits differ among individuals of the same species. So these are two different genetic traits that we can see in a species: invariant and variable traits. So these are again we can divide traits into three different kinds: physical traits, biochemical traits, and behavioral traits. So the physical traits are physiological characters that inherit from your parents, so like inherit from generation to generation. So these are easily observable, or you can easily see, such as like shape of a dog's face, or like the color, things like that. And biochemical traits, so these are like, it's hard to observe. So these are more like chemical reactions that happens inside the dog's body or any organism's body. So in this case, a dog's susceptibility to Addison's disease. So this is caused by a chemical imbalance inside the body. So this is a biochemical trait. And the third type is behavioral trait. So a trait that kind of like has to do something with the behavior. So like example, a dog's shyness or like extroversion. So these are some genetic features. So even the shyness is kind of like a genetic trait. So now let's look at some of the terms that we're gonna use in this chapter. So gene is the basic unit of information affecting a genetic trait. So we talked about this in previous lectures too. So gene is the basic unit that kind of like produce like a functional protein. So genes are present on your chromosomes. Okay, so if you remember, the chromosomes are the genetic material that you can find inside the nucleus, right? So and so chromosomes, they are stretch of DNA, and the chromosomes contain the information in the form of genes or in the form of code. So we call alleles the different version of the same gene. Okay, so for example, like if there is a different version, some genes have 2 copies. Okay, so in human, we are diploid organisms. That means for every gene we have 2 copies. So one from our mother's side and one from our father's side. So in that case, like these alleles or the same copy of the gene, they might be slightly different. So these different copies of the same gene we call alleles. So here in this picture, this is kind of like dividing from cells to the DNA. So here this is the cell and here's the nucleus. Sorry. So the inside nucleus, so these are chromosomes, long thread-like structures, and you can see these are paired because they are diploid organisms. So when you enlarge on each pair, you have these are single chromosomes, and in single chromosomes, so each band it represent a single gene, gene A. So there are 2 copies, one copy on a red chromosome, one copy on the blue chromosome. So that is representing one from each parent. And you can see this is showing up the DNA double helix. Okay. So now when we talk about a genotype of a particular character, so the genotype is the genetic makeup of an individual trait. Okay, so that means whatever the genetic composition of that trait, what caused that genetic trait, what is the gene composition that leads for that trait. Okay, so for example, like for the fur color of dogs, what is the genetic composition that leads for a particular fur color? Okay, and then the phenotype is the physical appearance of that genetic trait, what you see from the outside. Okay, so what you can observe from the outside. So that's what we call the phenotype. So the phenotype is what you see. Genotype is the genetic makeup of whatever you see. So I mentioned that earlier each gene has alleles. Okay, so that means alleles are slightly different copies of a single gene. Okay, so sometimes those different copies are have more power over the other copy. Okay, so in that case, if a particular allele is more powerful over the other one, we call that the dominant allele. And if a particular allele is less powerful than the other one, or has no effect on the over the other one, we call it recessive allele. Okay, so normally we use uppercase letters to represent dominant allele. For example, like if we talk about the black fur allele, we use uppercase B. Okay, and for that same color, recessive allele would be lowercase b. So when in a dog we have like black color phenotype, then it can be, the gene composition can be like this. So the gene composition can be uppercase B like this and lowercase b. Okay, so this would be the genotype. Okay, so in this case, this uppercase B is the dominant allele and this lowercase b is the recessive allele. So the dominant means it is more powerful when these two present, and the recessive means this copy doesn't have has no power over the dominant allele. So now what do we call homozygous homozygous genes, and what are the heterozygous genes? Okay, so when both alleles are same type, like this B uppercase BB or lowercase bb, so we call this homozygous. So in this case both copies are dominant allele, uppercase B, uppercase B. In this case the both alleles are recessive allele. So that's what we call homozygous. Okay, so in this case the dog will have a black fur color. Okay, and in this case the phenotype would be kind of brown, not black. Okay, the heterozygous means the both alleles are different from each other. So it can be like uppercase B, dominant allele, and lowercase b, recessive allele. Okay, so when a dog has this combination, that dog will have, because it has this dominant allele, that dog will have a black fur coat. So lots of geneticists or scientists that study genetic, they work with the soybean plant. The reason is like when they use soybean, the generation time is very fast. Okay, so for example, if we do research with dogs, normally the lifespan of a dog is about like 12 to 15 years, right? But if you use soybeans, you can like get multiple generations within 5 to 6 months. Okay, so you can get like you can cross the plant, you can like produce seeds, and then you can germinate the seeds and then get the next population. So you can have multiple generation very fast when you use these plants instead of like animals. Okay, so that's why lots of geneticists prefer plants in their experiments than using animals. So one of the very famous geneticists or scientists that worked on genetic is called Gregory Mendel. So he used pea plants to study what happens to gene in different generation. Okay, so he's actually called father of modern genetics, and he used pea plants, and then he cross-pollinated and then see what happens to their flower color and what happens to their seed, like seed color, and lots of phenotypes were analyzed and to figure out what's happening at the genetic level or the gene level. So these are some of his findings from these experiments. So here he used these different colored flowers. So here he used purple color pea plants, purple color flower pea plant, and white color flower containing pea plants. Okay, and then he cross-pollinated those flowers, and the next generation, so the parent generation is pea generation. So the first generation he called F1 generation. So he got the seeds, and when he germinated, all the flowers were purple. Okay, and then he got the seeds from these. He cross-pollinated the F1 generation, so that means he got the pollen from these plant, and then put it on the egg or like kind of ovule on the another plant, and collected seeds and put them on pots. And when he did that in the F2 generation, he observed every out of 4 plants, every 1 plant had white flowers, and 3 plants out of 4 had purple flowers. Okay, so now let's see like why that happened. Okay, all right. So here's what happened. Okay, so in the first generation, so all right, so in the first generation, so this is the parent plant. Okay, so this is a parent plant, and this is purple. Okay, so in this case, let's start with PP. Okay, so this is the parent plant, purple. Okay, and this one white, so the white is the recessive phenotype, so I'm going to use lowercase p, just like this. Okay, so when he cross-pollinate these two, whatever the F1 generation is going to be heterozygous, so that means uppercase P and lowercase p, like this. Okay, and when he cross-pollinate these two together, uppercase P, and so this is again is gonna be like this. Okay, so then he cross-pollinated these two like this, and then he got this variety. Okay, so we will use Punnett square to find out. So we definitely know this is lowercase pp, because in order to be like white, it needs to be recessive. Okay, so this is white. Sorry. Okay, excuse me. All right, so just remember the parent generation P, so the next one F1, The next one is F2. All right. So a Punnett square is a technique that we can use to find or the predict offspring genotype and phenotype if we know the parent genotype. Okay, so here you just draw a kind of like a box, and on top you put the parent's sperm and egg. Okay, so in this case, the F1 generation you have this heterozygous cross. Okay, so like this P and P, and then P and P. Okay, so this one when you cross, so the uppercase P and little p. So this one uppercase P, little p. So when you have these gametes, so this one and this one, uppercase P, so they will come together to form this one, and then this P will join with this one, so this is the next one, and this one will come with to form this one, so this is P and P, and finally little p will join with this little p to form lowercase p. So this is the white flower. So 3 to 1 ratio, purple to 1. Okay. So in this way we can predict the offspring if we know the genotype of parents. So now, so depending on these experiments, Mendel came up with some laws of some different laws. So the first one of the laws he came up with is the law of segregation. So in this law, he mentioned that if the two alleles of a gene are separated during meiosis 1 and can end up in different gametes. Okay, so that means they can segregate evenly. So any gamete can go and join with the other opposite gamete from the parent. Okay, and here, so pretty much if you know the probability, so if you have the possibility of 0.5, so if you toss a coin with heads and tail, the probability of getting head would be 0.5, or like a half 1 to 2, like a 1 out of 2. 1 out of 2, so like 0.5%. So it's the same as like getting a probability is like the same as like the like segregation theory. So now if we look at like in the second generation, so according to the Punnett scale, so getting a black coat is the chance is 0.75. So if you have offspring like double uppercase B, little b cross with uppercase B, little b, and if you remember out of you remember 3 to 1 ratio, so 3 to 1, so 3 is going to be black and 1 is going to be brown, gray or brown or gray. So the chance of getting a black dog is 75% chance, or 3/4. Okay. So and then so the law of independent assortment. So this one explain like when aligned with the alignment. So we discussed this during the last lecture too. So during the meiosis, when the chromosomes align in the center, the chromosomes has the opportunity to align freely on the center, so they can kind of like segregate into either sides of the cell. So in that way they have the chance to go like segregate into any of the eggs. Okay. All right, so like this. So when the eggs are formed, these different alleles, they have the chance of like getting into any of the combination. You can have multiple combination and getting into any of the egg alleles together. Okay, so here you are now looking at two different genes, how it segregate. Okay, so in this one, uh, the scientists look at the shape of the pea and also the color of the pea. So the yellow and green, and round versus wrinkled. Okay, so the yellow is dominant versus green. So you can see the yellow has uppercase Y, green has the lowercase y. So in order to see the green color, you need to have two lowercase y. Okay, and also the round color is the dominant allele. So you can see in order to see wrinkled peas, you need to have both alleles in the lower case R and R. So both R should be lowercase in order to see wrinkle. So in order to see wrinkle and green, so this is the genotype. Okay, so like this. So here this one will create this allele, capital R, capital Y gamete. So this one will create this gamete, little r, little y. So when these two interact or fertilize, this is what we're gonna get. We're gonna get a yellow round pea, and then when this one when this plant get germinated, and it's going to produce various different gametes. So these are the possible gametes it can produce. Okay, so then these gametes can get self-pollinated, and these are the options. Okay, so you can see like out of 16, so 1 2 3 4 4 times 4 16. Out of 16, 1 is going to be wrinkled green, and then 9 is going to be round yellow. So round green 3, wrinkled yellow 3. So 9 to 3 to 3 to 1. So that's the ratio. Okay. So Mendelian traits, so we can actually like use multiple genes and look at how they kind of segregate together, as long as they are not on the same chromosome, and look at like how they kind of behave, control phenotypes together. And like here the myth is eye color is a simple genetic trait and blue eyes are recessive. So the fact is at least 12 genetic genes contribute to the eye color, and blue eyes are not determined by a recessive allele at one gene. Two blue-eyed parents can in fact have brown-eyed child. So as you see, the eye color is not controlled by a single gene. There are at least 12 genes contribute to eye color. So there are multiple genes that control a single trait. so the chromosome theory of inheritance that explains the mechanism underlying Mendel's law. So chromosomes are paired, and each homologous chromosome in a pair has one allele for a gene, and alleles are shuffled and recombined and then separated randomly into sperm and egg cells during meiosis. During fertilization, a 1 in a million sperm fuses with 1 in a million egg to create a unique individual. So back to our dog story. So there are lots of different characters that we can see in dogs. So and there are lots of research done in dogs. So you can see this is a shadow, a standard poodle. So this is the first dog to have its genome partially sequenced, about 80%. So this is a Tasha, a boxer. So this was the first dog to have its complete genome sequence. And so the boxers are vulnerable to hip, thyroid and heart problems. So, and these are some of the more information. And we know that lots of genetic disorders are common in humans and dogs, such as cancer, epilepsy, heart disease and Addison disease. They are common diseases to both human and dogs. So incomplete dominance is when neither allele is able to express its full effect. So for example, heterozygous displays in an intermediate phenotype. So in this case, genotype LL. So in this case, phenotype large. So in the the genotype, it is heterozygous, but none of the alleles can be is over dominant over the other. So that's what we call incomplete dominance. And this one again, BB is small, so these are kind of controversial these alleles, they are not completely suppressing the other. Okay, and this one is medium. Okay, all right. So here actually the large one, so this is the homozygous LL. It shows the fully large one. So the BB, so this is the small one. And then the medium one, so this one is kind of not resistant, so it's in between these two. So that's what we call incomplete dominance. So codominance is something two alleles are equally visible in the phenotype of the heterozygote. So for example, gum color in dogs, pink and black. So if the pink PP, black uppercase PP, dominant allele. So if the pup has uppercase P and little case P, they both have pink with black spot. So that's what we call codominance. Both alleles show up on the pup's gum, and this is similar to our blood type AB in humans. So complex traits are the genetic traits whose pattern of inheritance cannot be predicted by Mendel's law of inheritance. So these do not fit the straightforward single gene, single phenotype pattern discussed so far. So, pleiotrophy is when a single gene influences a number of different traits. Okay, so for example, the shape of a dog's head and the shape of its limb bones are controlled by a single gene. In silver foxes, tamer foxes also had floppier ears, shorter legs, wider faces, and curlier tails. Okay, so in this case, like single gene controls multiple characters. So polygenic traits, so single trait governed by more than one gene. So examples in human: eye color. So more than 12 genes control the eye color, skin color, body size, running speed, body pressure, blood pressure. These are like kind of controlled by more than a single gene. A gene can have more than 2 alleles, but each individual can carry at most 2 of them. So in humans, ABO blood type has 3 alleles, A, B, and O, but each individual can carry 2 of them. So epistasis is what we call like when 2 genes are inherited independently, but the allele of one gene affect the expression of the other gene's allele. Okay, so for example, coat color of Labrador retrievers. Okay, so for example, B and E. So when like when the B when the E present, okay, it affects the expression of the other gene. Okay, so in that case, like no matter like you have whatever the other gene, you get kind of brown color. So here like in the without even like when you have little e, that's the Labrador with yellow color. So if the environment affects the phenotype, it becomes nearly impossible to predict the phenotype when given only the genotype of the individual of his parent. Okay, so for example, some genes are affected by the environment. Okay, so for example, like in this Siamese cat, the ear color is kind of like changed due to the temperature. Okay, so for example, like the cold extremities, like kind of like the ears and the paws, they are like darker compared to the core, like the body. So wherever the core body parts are whiter, and these extremities, they are kind of darker. So most of the chronic diseases have complex traits. So for like colon cancer, stroke, heart disease, diabetes. They have complex traits, and you can see like higher rates of animal death caused by these diseases in the US. And so the complex traits, we know that the common diseases in industrial countries caused by multiple genes interacting complex with each other and external factors like heart disease, cancer, stroke, diabetes, asthma, arthritis, chronic diseases that people have for the rest of their lives. So major goal of modern genetics is to identify genes contributing to human diseases. So, such as like what are the genes all kind of alleles that control high blood pressure, heart disease, diabetes, Alzheimer's disease, and some cancers, schizophrenia. And genetic test tells us if we are predisposed to diseases before we become ill with it. So we can like kind of take preventive measures to reduce the chance of developing these conditions. And also like with this research on dogs do the same thing. We can kind of take care of the dogs as well as like kind of uh, um, identify the genes and see whether there are any kind of factors that affect for these diseases. All right, thank you.